I'm Julien Baruteau. I work in London in a pediatric hospital, and I have a clinical activity looking after patients with inherited metabolic diseases, and as well a research group working on designing new treatment for rare genetic diseases. So I have some funding from the foundation. This is my disclosure for this talk. So I wanted to start with what is Citrin. And the first thing is that Citrin is a protein, which is a transporter. So that's a delivery system like any other delivery approach, which is bringing something in a different location. So from London, we have this tower bridge, which can open to let some boats go. And you have an open configuration and a closed configuration. When your transporter doesn't work or is closed, you have issue with transport. This is the very basics. So this transporter is enabling the exchanges between different biochemicals called glutamate and aspartate, which are going in different directions, in opposite directions. And these exchanges are being between different locations in the cell. One is what we call the cytosol, which is the yellow area that you can see. And the other aspect is the mitochondria, which are the batteries or the power plants of the cell, which are generating the energy. And these biochemicals are important with the Citrin transporter sitting really at the membrane of this mitochondria to be able to exchange biochemicals between the inside of the mitochondria and this yellow area that we call the cytoplasm. So why is this transporter Citrin so important? In fact, it's connected to many important pathways in the cell, in the body. So this is a lot linked to different, it's linked to energy, for example. So you can see here that we need Citrin in order to use the glucose to make energy from glucose or to synthesize glucose. We need energy, we need Citrin for making energy from fat. We need Citrin to generate the energy from inside the cell, from these mitochondrial organelles in the cell. We need Citrin for a specific pathway, which is called the urea cycle, which enables the detoxification of a toxic compound, which is called ammonia, and the biochemical reaction, the biochemical composition is called NH3. And we need as well the Citrin transporter for detoxification of alcohol. So what I've just showed you can be summarized in a very complex way, in a different type of complex structures and transporters and reactions. You can see all the different transporters which are linked to on the mitochondrial membrane, which are as well in the vicinity of Citrin. So this is a quite complex system, but Citrin is really at the center of this aspect and is involved in all of these reactions. So when we look in more details, so we can really summarize two different roles of the Citrin transporter. It's involved in energy production, and it's involved as well in detoxification in general for this toxic ammonia and for alcohol in general. So these two aspects are really essential in the role that this transporter has in the body. Just a few words about the urea cycle. This is a liver-based pathway which enables the detoxification of this ammonia compound, which is protein by a constant turnover and breakdown of proteins. And it's supposed to convert this toxic ammonia into urea, which is a non-toxic biochemical. So this is a really an important pathway, and because we produce in our body just by degrading proteins around 10 to 20 grams of nitrogen, which is an essential component of the ammonia per day. And all the proteins involved in this cycle represents more than 5% of the total liver mass. So that's quite a lot. And this is a process, a cycle, which requires quite a lot of energy because it consumes three molecules which we call ATP, which is really the essential energy molecule produced by the mitochondria to detoxify two molecules of ammonia and produce one molecule of urea. So when this pathway urea cycle is impaired, is deficient, the ammonia levels will build up in the body and will cause some acute neurological symptoms, some insult of the brain, with some sleepiness, with some confusion, with some imbalance, with some coma, for example. And this is really reflecting the severe insult that this ammonia can have on the brain. On the low right, you can see a brain MRI of a patient who has suffered some severe hyperammonemic insult, so insult of ammonia on the brain. And you can see that the what we call the ventricles, which represent this area full of liquid inside the brain, are quite dilated. And this is because the size of the brain, the parenchymal, has reduced, what we call an atrophy. So that means that the number of cells in the brain has decreased, has been lost because of the insult caused by ammonia. When we discuss about citrin, we discuss a lot as well about a couple of other small biochemical compounds that we call NAD and NADH. So what is this? NAD stands for nicotinamide adenine dinucleotide. This is a large molecule, which is what we call a coenzyme. That means that it's helpful for some specific enzymes, some specific seizures in the body, which will help to transform some biochemicals. And this is an essential biochemical which will help accelerating this reaction. So if we can really have a simple understanding of the role of this small molecule as some rechargeable battery. So you can think that the NADH might be a sort of charged little battery system, and the NAD will be an empty battery. And so if you think about the application of this into the citrin pathway, so we have, for example, the glucose that will be useful to produce some NADH, so this charged battery. And what we want is to pass this charged battery across the mitochondrial membrane to generate some energy, what we call ATP, in the mitochondria. In fact, the mitochondrial membrane is impermeable to NADH, so we need a system that will be emptying this NADH into NAD, emptying our battery, and use biochemicals that will be passing through the mitochondrial membrane to enable to cross this NADH into the mitochondria. So the citrin is very much at the center of this pathway. And if the citrin doesn't work properly, effectively, we can't really have this NADH crossing, and we can't have an effective production of energy. It's very much like a electrical current going through and trying to light a light bulb in the mitochondria. If you have the switch which is off, like the citrin transporter which is off, you will not be able to pass the current, pass your energy in the mitochondria, and you will not be able to produce enough energy. So what is citrin deficiency? So this is a rare disease, and we speak about rare disease when we have one affected person which is affected in more than 2,000 people. The prevalence of this disease, where we can find more patients in the globe, is very much in East Asia, and the country in which we have one of the highest, more important population of patients is Japan, when you have 1 in 17,000 people affected by the disease. So this is a genetic disease. That means that we have abnormal mutation in the DNA, in a specific gene, which is called SLC25A13. And we have different type of misspelling, different type of mutations that I've shown you here. So if we have, for example, a little word which will represent some DNA sequence, it's like a word with different letters, and you have some abnormal letters which are being either inverted, changed, or deleted, for example. And that is what is causing the abnormal expression of this protein, of this transporter from a misspelled altered gene. So this is what we call a recessive disease. That means that both parents will be carrier of the disease, and there is one risk out of 4 combination that these 2 faulty copy of the gene could be together and will generate then the disease. So how is this NAD system working in CD, in citrin deficiency? So if your citrin transporter doesn't work, so you can't move your aspartate into the cytoplasm, so you have a deficit of this compound called oxaloacetate, so you can't generate NAD from your NADH, and then you have all the consequences across. You can't generate then some NADH in the mitochondria, so in fact you don't manage to synthesize enough ATP, enough energy in your mitochondria. So if you don't have so you have too much charge NADH in the cytoplasm, and you don't have enough in the mitochondria. So you have this deficit, this imbalance, which is causing which is why this NADH NAD ratio is a marker of the disease in that. So you have that's like a closed bridge, a closed transporter. So what are the signs of the disease? That's very much relying on the mechanism that I've just explained. So if the different pathways that I've shown you before do not work, if you can't, for example, use your glucose properly, you will not be able to generate enough energy from the glucose, and you will have potentially some symptoms triggered by carbohydrates, for example. You will have difficulty in regenerating glucose as well in your cells, so you may be at risk of hypoglycemia, so low glucose levels in the body. You will not be able to degrade properly your fat, so you will have an accumulation of fat in the liver, what we call steatosis. You will be struggling to synthesize some energy in your cells, so it can generate to some fatigue. You will not be able to detoxify your body from ammonia, so you will have what we call hyperammonemia, and you will not be able to detoxify properly alcohol from your body as well, so you may have some alcohol-triggered symptoms. So the particularity of this disease is that you have some age-dependent presentation. So you will have, for example, in the newborns, a jaundice, which is quite characteristic, with an accumulation of bile acids in the liver. So this can be linked to a yellowish coloration of the skin, white stools, poor growth, low blood sugar levels, high ammonia levels. When usually the patient will be recovering gradually from this specific neonatal presentation, and then there will be what we called theoretically a silent period where there is less symptoms. So the patient might be having some dietary preferences, avoiding carbohydrates, which can trigger some symptoms and discomfort, preferring some proteins-based or fat-based food. The growth can be quite short. There is potentially some fatigue, some abdominal pain, and some low blood sugar levels and high triglycerides, which is some high fat in the blood. So I think the silent period could be put into brackets because it's not such a silent period, and there are still significant number of symptoms we can see at that age. And then there is in adulthood some potential liver complications we can see, fatty liver, and more pronounced symptoms with some scarring tissue in the liver, what we call cirrhosis, potentially some liver cancer in some cases, and some high ammonia with the consequence on neurological symptoms. The presentation in adulthood has been called previously citrinemia type 2. It's called now adolescent and adult citrin deficiency. It's not seen in all patients, maybe around 1 in 10 patients, and it can be seen from adolescence to late adulthood. So I mean late adulthood meaning probably above after 60. It can start quite abruptly, and it's more frequent in male compared to female. So there is a ratio of 2.5 to 1, and it's often triggered by some consumption of alcohol, carbohydrates, some medication, some fasting, etc. And it can happen with or without prior symptoms. So, for example, some patients will be presenting with this late presentation without having had any symptoms in the past, no neonatal presentation, for example. The neurological symptoms associated with the high ammonia levels can be quite variable, from confusion, abnormal behavior, memory issues, seizures, sleepiness, going up to a coma, and this chronic liver disease I was mentioning before. So this is an example of pictures of what we can see in terms of liver disease in this condition. We discussed about fatty liver. So that's, if you look on the left side, you can see some arrows showing some whitish bubbles. This is the accumulation of fat in the liver cells, what we call steatosis. And on the right side, you can see some blue tissue, a little bit fibrous. That's the scarring tissue that I was describing, what we call fibrosis, which can evolve towards cirrhosis later on. So how is the disease diagnosed? So that's a combination of a compatible medical history, so compatible symptoms which will fit in the disease presentation, some blood tests which could show some of the classical signs of the disease. So, for example, low glucose, high ammonia, high triglycerides, in some cases high bilirubin. We can have sometimes some abnormal profile of amino acids, high citrulline, which has been giving the name of the late presentation previously, citrinemia type 2, and we have other abnormal amino acids which can be seen as well. But what is really making the diagnosis is very much the genetic analysis of the gene, which will be able to pick up two mutations, which will be effectively signing up for the diagnosis of citrin deficiency. So what is the treatment that we have? At the moment, the treatment is very much relying on a diet. And that's why when we look after patients, that's very much a combination of a treatment where we need a dietitian to help managing the patients the best way possible. So in the neonatal period, when there is this episode of jaundice, we will try to exclude or limit the amount of lactose in the diet. We will try then to keep medium chain triglycerides being given at all ages, because there is various benefits in using this specific already mildly broken fats in the diet. It can increase the production of energy. It can regenerate the ANAD in the cytoplasm. It can facilitate the release of fat from the liver. So it has different, as in different ways, this can help the symptoms of the disease. So most of the patients will have some degree of aversion for carbohydrates and a certain preference for proteins, and this is reflected by the composition of the diet of the patients. You can see that the amount in percentage of protein is slightly higher. The composition of fat is slightly higher as well, and the amount of carbohydrates is slightly lower compared to a standard diet. So there are some measures that are being considered depending on the symptoms of the patients, but as a rule, we try to avoid as much as possible some glucose infusions unless there are some low blood sugar levels, because this can be potentially triggering some symptoms. We try to avoid alcohol as much as possible as well. We can, if there is an increase of ammonia levels, propose some drugs which will be chelating, reducing the levels of ammonia. And we can consider as well some bile fluidifier, some treatment which will help removing the accumulation of bile acids in the liver. The liver transplantation is today the only curative treatment which will be proposed when we have repeated high ammonia levels, which are difficult to stabilize, and when we have a presentation with a severe liver disease, which will be either a cirrhosis with a scarring liver, or when we have, for example, a liver cancer potentially favored by this cirrhosis. So what are the new treatments on the horizon? So we discussed about the medium chain triglyceride that you can see in purple. You can see there are different options that are being studied and considered, which are all here in red. Some of them are based on some dietary supplements. Some of them are based on small pharmaceutical molecules. Some of them are based on some specific ways of doing gene therapies. And so can just have a quick outline of some medications that are maybe more considered a little bit further up in the development or being studied through projects which are being funded by the Citrin Foundation. One is the nicotinamide ribosides that's being considered to try to regenerate the NAD in the cytoplasm by giving some supplements by these compounds, which is a food supplement. And then there are some different approaches which are being worked on with different type of gene therapies. So you can see that to make a protein, we need a DNA, a gene, which will then be producing what we call a messenger RNA, which is in fact giving instructions to the cells on how to make the protein. So we could consider either a messenger RNA replacement approach, where we give some messenger RNA directly to the cells which is impaired, and so that will give instructions to make the citrin transporter. Or we can do what we call mutation editing, for example. So we'll be using some molecular scissors, which will be correcting directly on the DNA the mutation, in order to then have all the right instructions to make an efficient citrin transporter. Very briefly about the models of the disease. There are different types. There are some liver human cell types which have been developed and which are being used to understand better the mechanisms of the disease, which is very much centered around the liver. And there are some citrin deficient mice. There are different models as well, which are not all perfect, but which can give us some insight, and is really a way to study as well the disease and potential treatments. But no model is perfect, and every single model has its own challenges. So what are the challenges in this disease? I think this is a rare disease, so every rare disease has some difficulties to try to have, for example, a good description of the disease, because there are few patients, especially when there are some variability across sexes, for example, across ages, across dietary habits in different countries. There is some heterogeneity, what I was mentioning before, in terms of signs according to the age of the patients. There is not a full understanding yet of all the disease mechanisms, and there is still some mysteries around that disease that we are really trying to address in different ways and with the support of the foundation. And there is still some limited information of some aspect of the disease and the symptoms that the patients are presenting. So there's still some work to be done to have a full picture of what's happening in this disease. What are the strengths? I think this is amongst the rare disease. This is not an ultra rare disease, certainly not. This is probably one of the most frequent rare diseases in the field of what we call inherited metabolic diseases, especially in Asia. There is a rapidly growing patient community, and I think this is a very important aspect that researchers, regulators, physicians are very much taking into account. And there is the support of a strong foundation, which is actively building a network of researchers, physicians, but as well patients around that disease, and creating really a powerful ecosystem to progress further in the treatment and better understanding of the disease. So in conclusion, this is a complex disease. What I hope I have illustrated that through my talk. This is a rare disease with its own challenges, and there is this growing and active community as well. And I think this is very important that patients and families can really drive the way the research is being set up. And this is very important to have this joint discussion and forum so that we can learn as researchers and physicians from the patients. Just like to have a very brief thank you to the patients and families that we learn from the research team, the hospital, the team in the hospital who is helping us, and the foundation for their support as well. And thank you for listening.